kind. Let's go ahead and invite Professor Anduza. See if the invite link works this time. And then, let me get the paper ready too. Y'all, the paper we're chatting about tonight, it's really... Thank you, Rand. Appreciate you. I'm really excited about chatting about this because last week we had kind of come to what was a really nice conclusion of what I thought was on the idea. Hi, Lobster Duck.
Hello there.
Of species and what species are and the living fossils and all that information. And then, sure enough, there's this paper. And now I think challenges it. Send it. Yep, you got it. Let me know, Danny, when I should send it. And so this paper, I think, very much counters and fights against the idea of what is the living fossil. Go for it. All right, you got it, Danny. Invite sent. Because this is looking at it from like a molecular genetic level, and the papers we were looking at last week were not looking at it on terms of genetics, but they were looking at it more of like structure. And so I'm really curious to see what conclusion we come to today. If this is something of a living fossil, are there exceptions to what living fossils can be? Maybe it's still not a living fossil. Either way, it would be a cool one. MJ, actually, Danny's the best one. Also, I hear Munchkin is home. Munchkin is upset. Munchkin just came home. She might make a visit, I don't know. Alright, hi Danny.
Hey there, Blint. Can you hear me?
I can hear you. Can you hear me?
I can. Excellent. So there was a weird glitch that was happening there, and I figured out how to, like, exploit it, and I'll leave it at that, because shoot. I might have to talk to, like, Twitch staff about that, because it feels like a very, I don't know. Something went wrong in a way that it shouldn't have.
Uh-oh.
But yeah. Anyway, I'm glad to be here. Some quick thinking, I was able to, anyway, apply some hacking there to get it to work. But, yeah. Anyway, it's good to see you. I'm not going to elaborate further.
I have to tell you, we wanted to go this weekend to see the Hadrosaur statue that you pointed out, but the weather, it was rainy. And so
I don't think that statue is going anywhere, so I think
I know, but I
think it'll be there the next time you go. Yeah.
Last week, y'all, we were talking about the Hadrosaur, and it turned out that there was a Hadrosaur statue. Danny's like, "Oh yeah, it's in Haddonfield." I'm like, "Excuse, sir, we live right next to Haddonfield." And
I had no idea, Blint. That is where dinosaur paleontology got its start in the United States. Like you were living in a place, or near a place, where like an entire branch of science saw its origin. That's pretty darn cool.
It's really I told Lita, and she was like, "No," and I'm like, I showed her the picture. I'm like, "This is here." And she's like, "We gotta go." So the plan is now we're gonna try this weekend to see if the weather will be better. And I know, like you said, it's not going anywhere. But I just want to go. No, Heox, it's in New Jersey. It is
Haddonfield, New Jersey. Yeah, Jersey. It's where dinosaurs got their beginning. You know.
It was I was I'm shocked, and so it's a Hadrosaur statue, and so we're gonna take
Hadrosaurus falkii. Named after the Falk family, I believe. Was it Peter Falk who discovered it? And it was from the marl pits of New Jersey, right there. I mean, that's one of the very first dinosaurs ever described from North America. The first dinosaur skeleton ever mounted anywhere in the world. You know, like when you go to a museum and you see a dinosaur skeleton standing up in a museum, the very first one in the entire world was there at the Academy of Natural Sciences in Philadelphia. And that was from the marl pits in Haddonfield, New Jersey. Yeah.
Was it one that was the actual dinosaur, or was this these one of these where it was pieced together from a bunch and the posture was all wrong? And
It was one skeleton.
okay,
Yeah. Yeah. So the
that's a
thing is,
bust.
yeah, that one specimen was from marine sediments. So we were talking about this the other day on my stream, where occasionally a dinosaur would die and then fall into a river and then get washed out to sea and then get deposited like at the bottom of the ocean at the time. That's one of those. It's from a marine marl. It's like marine sedimentary rock. Probably the same kinds of rock that you're looking at when you go out fossil hunting, Belinda.
Oh,
So you
nice.
find a lot of ocean animal fossils there.
Yeah.
Yeah. Yeah, it might be those same deposits.
Cool.
It's cool stuff. Yeah. Yeah. Remind me, and I'll send you a video about that with Ted Deschler talking about that and talking about New Jersey's importance for the earliest history of dinosaur paleo in North America.
Yeah, that would be really cool. I never made the connection that and when you mentioned, I'm like, wait, he can't be. Put it in Google Maps. Eight minutes away. Like, lobster duck, you didn't get us.
Anyway, how are you doing, Belinda? You have not fallen down. You've not fallen down the
into the wet basement.
Yeah. Yeah. How are things? How you doing?
We the basement now has a bunch of holes in it. The drywall they've cut out, so that's at least removed.
Yeah.
So that, you know, it's a little bit it's cleaner on that. You know, you deal with the insurance now. I was on the phone with them today. Like, all right, how are we gonna fix up the downstairs? Fishman, sleep well, my friend. Thank you again for hanging with us in the raid earlier today. What about you, Danny? So today was a world not hippo. Goodness, we were talking about hippos earlier.
Manatee
Day.
Appreciation Day. Yeah.
How was it?
Yeah. It was good. I don't know how much you're probably not able to catch much of it, Bullet, but what out of all of the mammals in the world, which mammal do you think might be closest related to manatees and dugongs?
A hippo.
That's a good guess. Well, it's a good guess in a certain way because hippos are the closest living relatives of whales. Manatees, however, and dugongs and their relatives are a totally different group. They are way over on the other side of the placental mammal family tree. Do you want to take a guess again? It's a big gray animal.
It's not an elephant, is it?
It is.
Really?
Yeah. Manatees and dugongs and the other sirenians, manatee-like animals, they are most closely related to elephants. And you can kind of see that when you recognize that, like, that tough gray skin that manatees have is homologous with elephants. And also, if you look at a manatee's hands, its front flippers, they've got little nails on them, like fingernails, and they look very, very, very similar to those of whales. Or no, whales, excuse me, of elephants, excuse me. Ugh.
So, what do you think the evolutionary process there was? Like, if they're closely related, like, was the common ancestor, we assume, was maybe aquatic when the divergence happened?
It's not. Shoot, you should look up an image maybe. I'll tell you what to search for if you want to. So, Afrotheria, AFROTHERIA, Afrotheria. It is a group of animals that are, we were talking about this today, they are really wild in that there's a bunch of different critters that you wouldn't think are related to one another, but they absolutely are. And that's because when, in the late Cretaceous period, about 80 million years ago, something like that, maybe 85, 90 million years ago, Africa had become this island continent off by itself. And the mammals that lived there during the time of the dinosaurs started to diversify, and for the next 70 million years, Africa would be an island continent without much influence from the outside world in terms of mammal evolution. So this one group of mammals that lived there, the Afrotheres, developed into all of these crazy different morphologies. You had ones that evolved to eat insects, like aardvarks. You had ones that evolved to eat plants and grasses, like elephants. Some took to the seas, like the sirenians, the manatees, and the dugongs. and then you had ones that evolved to look like moles. You had ones that evolved to look like shrews. You had just all kinds of crazy different afrotheres evolving into different niches just because they didn't have competition from these other groups of mammals. And then later, some of those spread out across the world, like the elephants and their relatives. Like mammoths used to live not just in they spread up into Asia and Europe and then North America, and some of them went down into South America, the other proboscideans. And it's really cool stuff to think about, just like how evolution and isolation can produce these really weird, cool critters.
Was this the question that was about the RNA?
It was, yeah. So that was the thing, and thank you for answering that, by the way. Apparently, this group, Afrotheria, was first proposed in a paper in 1998.
Huh.
And it was, I guess they were looking at RNA at the time. They were trying to figure out the mammal family tree, looking at RNA for like the first time, I guess. And suddenly, this group of critters that nobody thought were closely related to each other because they look so different, their RNA kind of matched up. And once we realized that, paleontologists took a closer look at some of their morphologies, and it turns out like there are little hints in their anatomy that show that, oh shoot, they are related, aren't they? So yeah, does that make sense?
Yeah. So in thinking about it, so my quick answer to that question,
Yeah.
we actually talked about it here too, because you came in and I typed back on your stream and everyone's like, well, wait, what was the answer? So my suspicion, right, is just ease and price. So at the time, like, sequencing was super, super expensive, and we still had the hypothesis that there was junk DNA, right? That there was everything that was in an intron was useless, and a lot of people were just comparing the exons, which is when you make the messenger RNA, originally you have all the exons, but then it's spliced down and removed, so you just are left with the coding sequence. And so they were probably comparing, the coding sequence, and so it's less to sequence because you're just looking at like a much smaller part of the genome.
That was one of my questions for today that I'll get into later about what is an exon. You don't have to answer it right now.
Oh, and I'll draw it out too. I'll draw it out as well. Sleep well, Turgus. And this Charlie's Dragon said this is the proposed image of the elephant manatee common ancestor.
Yeah, I think that's. I don't know if that's a common ancestor between elephants and manatees. I think Meratherium comes later. Yeah, the ancestor would have been back in the Cretaceous, back when everything looked like a shrew, basically. But this is like, I think this is supposed to be Meratherium here.
Okay.
And that is like the Eocene of Egypt, I think. And that's proposed to be an ancestor of the elephants. But this would be long after the common ancestor of elephants and manatees.
So it might be probably along this radiation here already, right? Where the split point is here. It would be
at the base of what we call Tethetheria, which is the common clade for manatees, Sirenians, and Proboscideans, elephants. Yeah.
Gotcha.
Yeah.
Nice.
But Meratherium is thought to be an ancestor of the elephants, so they were definitely right about that. Yeah.
Gotcha.
Yeah.
Yeah, so the other thing is, too, the technology, I guess, for getting the RNA at the time was much easier because one of the catches is the DNA sequences are super long, and so you have to have a way to break them up and tag them. So the idea being, and you can pause me if it's not if it's too off topic, but just I think it's interesting as to maybe why they went with it. Let me move over my source so it's a little bit more visible. Let's see. Go. So when you're doing the sequencing, right? If this is the DNA strand, there's regions in there that are going to be coding so that they will convert to a protein. So let's say like this region, and this region. So if that's the DNA.
In dreams, one is
sleep well, Zedman.
by earthly limitations.
What does that mean?
Come.
The protein, right, eventually after all the processing is done, will be those 3 bits of blue.
Yeah.
And that'll make the protein that is important for whatever process we're going to be looking at.
When you say when you say when the processing is done, what does that mean? Processing? Does that mean scientists processing this in the lab, or does that mean that the animal is developing?
So in the body, so like in our cells, as we're
Uh-huh.
producing proteins, the general usual process is you go from DNA to RNA to protein. And the first
Okay.
step is to copy your DNA strand. So the intermediate steps here, if we make this, if we draw this out as it actually is, that DNA is double stranded, right?
Yeah.
We want to make a single stranded copy, which is then called RNA. Let me just, there we go, erase it. So then you make the single stranded copy, and there you still have all these introns in green.
Okay.
And those introns need to be removed in order to make your protein. So there are processing factors called the spliceosome. So once this RNA molecule is made, these regions in green are removed in the body. And during development, during us breathing right now, constantly happening to make this final product.
And then you're left with the exons, which are in blue, basically.
Yep.
Okay.
And
Okay.
For a long time, it was thought these green regions were this useless junk DNA, right? Because if they're removed, what's the point of them?
Who needs them?
Right.
Yeah.
And it turns out they're important for structure of the chromatin, and then for regulatory information. So, like, what genes are accessible when, these,
Gotcha.
these can connect with one another and close down certain genetic regions so they're not accessible. They can open up regions, so it's more regulatory features that are present.
Sure.
And so those regions can undergo, mutation just like the exons, and that can change how and when genes are turned on and off. But if the idea for sequencing RNA is, they're not looking for this population here, they're looking for this short blue one only.
Which is kind of simpler to look at and presumably, like, cheaper to do back in the 90s.
Yeah. Because it's,
Gotcha.
if you went from DNA as well, like, the process for DNA sequencing gets really hairy, because if you have this two long strands, you have to go in and break the strands, because especially with those tools, you need to amplify the DNA. You need to make a lot of it so that you can eventually sequence it. The strands are too long to amplify on their own, so you need to break them.
Okay.
And then after you break them, you need to put a tag onto the ends of them that's something unique, and then those tags are the things that are amplified. Because you know what the tag sequence is, and you don't know what the sequence is inside of the DNA molecule. So let's say the sequence for this is, AATT, you will make a primer that will connect to the DNA sequence that's the opposite, TTAA, so it's just the nucleotides. And then your primer will bind and you can amplify. And what's cool about this is if you know this green sequence that you're adding on to the end of all these strands, it doesn't matter what's in the blue. That can be unknown because you're just going to amplify whatever's in there. So as long as you attach something known, you're in business.
Interesting.
So it's a neat trick, but that trick was super expensive and super, super pricey at the time. Nowadays, it's getting much cheaper to do. The other thing that surprisingly influences it too is genome size. So some of those animals in that graph, they probably had different sizes of genomes, and that influences the price. And so it's like, it's something that we don't even think about. Like when I was doing these sequencing experiments, it's like how many sequences fit on a flow cell, which is the unit that you put into the sequencer. And so with ants, it was like, okay, given the genome size, we can put for RNA sequencing 26 samples. But if we were to do humans, we could do 10. Because there is a threshold of information that the sequencer can read. And so that adds to the price as well. So it's all these like unique features that are constantly coming in. Does that make sense?
It does, yeah. And I think what I'm curious about is when we're talking about price like this, and we're talking about numbers, how much do you think that sort of thing might have cost back in 1998 as compared to today? How much has the price come down, if you could throw out some numbers just off the top of your head?
So, the human genome was published in '01, I believe, and that was millions of dollars with 30 plus different labs working on it. So it, I would say it's probably like easily $1 million. Easily $1 million.
Wow.
And it, granted, right, the genome is large, the human genome relative to others, and they actually didn't get full coverage, like everywhere on the genome being covered. So there was only actually last year did we actually get all of the human genome sequenced, because there were regions, last year,
2023,
because of technological limitations, because there's regions that are so tightly packed that they're just inaccessible by regular technology.
Wait, but if that's the case, and presumably the human genome would be the genome out of all organisms on Earth that people would focus on first, does that mean that that's the only genome in existence that has been fully sequenced?
For humans, or just anything?
For anything. Like, I would think that if any critter was going to have its genome sequenced first, and absolutely, thoroughly, fully 100%, it would be humans. Does that mean that we are, so far, if it only happened last year, that we're the only organism with 100% of our genome fully sequenced?
So the much smaller and simpler things like yeast.
Oh, okay.
So for example, right, our I have this.
How many base pairs do we have, Belen? Yeah.
Well, so not even the base pairs, right? Like if our DNA was in a circle, it would be it would be super easy. It wouldn't matter how big it is. It's in a circle, right? There's no structural issue, but there is this technique called, hold on, let's see, chromatin painting, where you can paint the entire chromosome in a cell. And so this, I think, will give you an idea. I think it's a really, really cool tech, and you can do it with any animal. So this is in a human cell, the regions where the DNA and chromosomes hang out. And so what's wild is there's this 3D structure to it. And so instead of just being like circular, it's like in loops and patterns and nicks and divots. And in a cell, it even looks like they're in particular regions. And so, right, because whenever you draw a cell, at least when I do... Oh my goodness, we got a raid from JD Does Dev. JD Does Dev Raiders, welcome the heck in. JD, how was your stream? I hope it was incredible. If y'all don't know our friend JD Does Dev, he is a game developer extraordinaire, a musician. He plays the saxophone, and he actually uses the Godot game engine. And he gave a really innovative talk at a conference not too long ago, or it was a conference, JD, right? Using a video game. So it was a video game based presentation on how the workflow does. And so instead of slides, the character moved to the next scene to illustrate the points of the work that he does. And so he's been very like, in a beautiful way, integrating his game development along with the day job as well, to show that, it's kind of like how we do, Danny, science and art, like with your 3D modeling, doesn't have to be insular. But as soon as you bring in these disciplines that people are like, no, that shouldn't exist, it really is beautiful. Yeah, and JD made a platformer as a PowerPoint. Also,
very cool.
Shout out, y'all. JD Does Dev. Because he's being innovative, his workplace is punishing him and not paying for any conferences. So he's got a GoFundMe. JD, feel free to post it to promote the ability for him to go to conferences and promote this kind of innovative techniques. So we're, raising money for JD on the GoFundMe, trying to get him to TwitchCon as well to present on some of these points. So JD, my friend, if there's anything you'd like to share, you know the drill. Drop it in the chat. We'd love to have you share it. For the Raiders coming on in, my name is Balint. I'm 1/3 of Science Streams. The other 2/3 are my lovely wife, Lita, and our daughter, baby, Alona. We're research scientists with our PhDs in molecular biology. Today, we're joined by Professor Danny Anduza, single greatest SciCommer on the entire planet. And he's a real-life paleontologist, and today we're having our crossover, talking genetics and fossil science. And there is an insanely cool paper we're going to be chatting about that both Danny and I we're going to send to each other. We didn't talk about it before, but ended up the same paper we ID'd that we want to talk about today.
Yep.
Which I'm really, really excited about. So, Raiders, welcome in. Guys, the GoFundMe is pinned to the top of the chat if you want to help out our friend JD. The second message he posted is to all his links, all the socials. Please go check out our friend JD. Thank you again for the raid, my friend. Welcome the heck in. And hello, Jimmy. It's good to see you too, Jimmy. We were just talking about this unique phenomena about DNA organization in a cell. So in a yeast cell, there is but a circular piece of DNA. And it's just floating throughout the cell.
Real simple.
Really simple. So that's why having that genome sequenced is pretty easy, all things considered, right? Because it's not big. There's no structural nuance to it. But then this... sorry, go ahead.
What percentage would you say, like what fraction of that is that of the human genome, for instance? Is it 1/16th, 1/100th,
It's like 5% at most. Tiny, yeah. Oh, and actually,
wow. Okay.
about the prices too. Alex Vixen mentioned that the estimated cost was $1 million in 2007, $1,000 in 2014, and today it's $600.
That's not for a whole genome. That's for some percentage of it, right?
Oh no, the human, like an animal's genome, you can sequence for pennies on the dollar at this point. Yeah,
Wow.
like you can, like it even has come down, like even RNA sequencing in my time. So where it used to be like 4 replicates of an experiment, and with like an experimental control. So like 8 replicates would be like $30,000 in 2013, is now $10,000 today. Like still a lot of money, but it's like constantly coming down with the tech, which I think is.
Is there like a clearinghouse? Or sorry, go ahead.
What do you mean, clearinghouse?
Is there like a clearinghouse of data where you can look at these different genomes? Is there like a database for this online that you can access?
Yeah, so it's on NCBI, so PubMed. You can just put in any animal that you want, like the genus, species, and it'll spit out it's called a FASTA file. I would not suggest to anyone to open that on your computer because it's not meant to be opened. You can export it like in Python or R, and then get like an Excel doc with all the nucleotides. But that like FASTQ file or FASTA file that's in there is like meant to be like opened by a different program. It will crash your computer because it won't know what to do with it.
That's funny.
And that is totally by I may have accidentally done that, or it just killed the computer. But yeah, so like, so Danny, this is a human cell.
Okay.
The nucleus of the cell, and what they've done is they've painted each chromosome. So it's a technique where they make a mirror image probe to every chromosome, and that probe lights up a certain color. And so like this is the real life image that you see in the cell. See like chromosome 1, let's say, is red, 2 is green that they've colored, and then when you put it into a cell, it'll glow these colors because it'll bind to whatever chromosome that you've designed this probe to. So you can see the 3D composition of where the chromosomes are, and you can see it's not like how we drew in school where it's a circle and squiggles in there, but each chromosome like occupies a particular region, and that is partially due to those introns and how they interact to form that 3D structure.
I only see 20 as the highest number on there. I don't see 23. Maybe your chair is covering up part of it?
Ooh, let's see. It might be.
No, that's covering up 1. Interesting.
It might be.
Or maybe are some on the other side or something?
There might be some on the other
21.
side, yeah.
Yeah.
So I just did not, like, this is from Ting Wu's lab, so she is she's the wife of George Church, which I know you've talked about on your stream, the mammoth guy.
Yeah, colossal,
Yeah, so he does mammoths, she does this kind of chromatin painting, and bugs in space is another thing that she does. Yeah. So yeah, there is the X and the Y in here, so there's the sex chromosomes, y'all, but then there's still the one more we've got missing.
I did not know. Wait, so the X and the Y chromosomes are not numbered among the one, or are those Because I don't see 22 or 23. It's not like the X and the Y are 22 and 23, right?
No, so that the X would be 23
Okay.
or Y. So like the XY would be
Uh-huh.
that set, would be the 23rd
Gotcha.
set, and then there should be 22 in here as well.
Okay. Neat. Shoot, Bolin, I have so many. I could talk about this all day and just pepper you with questions.
But yeah, so
Um,
that's I think the cool part is that there is this 3D structure, and that's all by the introns, which we thought was junk DNA. Right, turns out is not actually junk DNA at all. It's all this like internal organization within the cell, which
Interesting.
I guess that's my rabbit hole into like these intronic regions that we drew out in the previous sketch is like why they're still important, and so sequencing them. It just depends what your question is, but there's still information to be garnered by between it. So, like, maybe our coding sequence to chimps and bonobos is really, really close, but those intronic regions can be different, and those contribute to regulation, and that contributes us to not being a bonobo, but being a human. Like, when and how those genes are turned off. And actually, the most prevalent example is actually a gene called MYC, MYC. It's a gene. It's a gene in cancer, and it's an extremely long sequence. Most of it is intron. Most of it is non-coding, and it turns out that when you get cancer, you don't get a mutation in the coding sequence that makes the protein. You get a mutation in the intronic region, which is the regulatory part, and that's what causes it to be overly turned on, leading to cancer. And I forget which type of cancer, but I think that 90% of all that class of cancer is some kind of mutation in the MYC regulatory region.
Interesting.
And so that was called junk DNA, as Dame Karen says. Like when I was in high school too, it was still junk DNA. But it turns out like that's where it's important to look. So back in the the 90s too, like as we're talking about the sequencing price, like a lot more of the genome is intronic than exon, and so it's just again price too. It's just there's technology like adding the labels and the tags, but then there's also just the amount of time, effort going in. So if you wanted to do a multi-organismal comparison, like RNA was much cheaper to be able to do. It is removing a lot of information, but
that makes a lot of sense.
Yeah, it just depends on what you, I guess, want out of it. It might, honestly, for the 90s, I wouldn't be surprised if they even just took a subset. Like, here are proteins that we're interested in, and we just want to compare what we think are important proteins between these animals, which is kind of what the paper we're going to talk about today, and just see how different they are. Because that could also be a proxy, right? Even if you don't get the whole, like, RNAome, you could still get that component and make a difference there, and just see what's important.
Is RNAome is that just a genome for RNA?
It's like it's officially the transcriptome.
Transcriptome. Okay.
So that's so the process right from the DNA to RNA is called transcription. So it's capturing everything that has undergone in that process. So it removes the DNA. So you're just left with different populations of RNA, and even there's non-coding RNAs. So to make it even more complicated, there's RNA molecules that don't make protein. There are RNA molecules that will bind to. If we draw it out here, if this is your DNA here, there are RNA molecules that will bind to DNA and prevent stuff from genes from being turned on. Because your transcription factor will try to bind here to turn on that gene and transcribe it, but if there's an RNA blocking it, then it can't do its job, and so it will physically block transcription from happening. So RNAs do a lot of things that are.
Where does messenger RNA fit into this?
So those are classically ones that become proteins.
Gotcha.
And even that isn't just straightforward as DNA, because the classic is you go from DNA to RNA, adding in the nuance of removing those introns now, right?
Yeah.
And then that exonic component becomes a protein. That protein isn't necessarily done when it becomes, amino acids. That also might get modified. So there's processes of glycosylation, so you add sugars onto it, or sumolation, or any so many molations under the sun that you're adding onto these proteins to further modify them. So it's not just like a one and done kind of thing. Like when you have the protein from the RNA, it's not necessarily done. So there's like further modifications that might also happen. But usually as we classically define, the messenger RNA becomes some kind of protein that does something. Then there's there's long non-coding RNAs that can bind to DNA and turn off things. There's small RNAs that can regulate proteins. So it's an RNA regulating a protein's population. So it's such a complicated picture, Danny, than what it was even 10 years ago. Like there's constantly there's papers coming out on new RNA species that are found, meaning that there's a new population of RNAs doing something that we never thought they would be doing.
Huh. That's wild.
Yeah. And
That's
and
really
a big
cool.
part of it is like, you might be like, well, Belent, why is it coming out now? It's because the technique didn't exist. The tech didn't exist to even test it. Like there's my favorite intellectual debacle that we come up with is that there's these populations, right, of small RNAs that are really, really tiny in size that we think are doing something. If you run them out on a gel, Philly, welcome in. If you run it out on a gel where you have positive charge here, negative charge here, DNA slightly negative, so your DNA will go down in the band and you can separate by size. Normally you'd want your sequences that you're going to sequence, let's say around here, for the sake of argument, in the middle of the gel, and you're like, oh, that looks really good. It's not too, if it was all up here, it'd be huge and you couldn't sequence it. So you want it in the middle. Okay, cool. Things that are down here, we have thought for decades are primer dimers, meaning that, remember, when we add these components onto the DNA to code them, so like we know exactly how we've broken the DNA, we've tacked on something and they can transcribe and copy the DNA. These things can bind to one another, and then that's something that you put in the mix. So you don't want to sequence that because it's just trash because you put it in, right?
Sure.
Well, it turns out that this population of supposed trash, yes, our stuff is there too that we added to do the sequencing, but then there's a whole population of RNA molecules there that are important in development, in behavior, in regulation of genetics. And so it's just like we don't even know how to get to them yet, really, because the tech doesn't exist. And so
Very cool.
Yeah.
Wow. This is mind-blowing. And I guess, I don't know. I know that I didn't know anything about genetics. But at a moment like this, you're really hammering that point home. Oh my goodness. Yeah.
Sorry, I got excited and carried. I was like, oh yeah, there's
this is really cool. No, this is really cool. I could listen to you all day about this.
And Golkanek is right that these kinds of observations do further solidify the idea that RNA is like that early life
Earliest
molecule. Yeah.
Yeah.
Because it also has like enzymatic ability. Like RNAs can interact with things as well. A little bit like how proteins can. They can also have
or viruses, right?
Yeah, exactly. Like prion regions that can touch and interact too. So it's not as simple as like, here's a molecule that makes protein. It's that it can make protein, but then there's all these regulatory elements as well, and it's just, and we're finding more and more things about it that people thought we were done 20 years ago. And so I think that is also why if you had unlimited money for doing the sequencing of that experiment, with the different animals that we were talking about with not this tree, but I may have closed that. Did I close the window? Shoot. With the tree from earlier. Did I close
the Afrotherians?
Yeah.
Yeah, yeah.
Shoot. Well, with that tree, that would have been the easiest thing to do. Like if you had unlimited money, is to sequence the entirety of the genome, get
Uh-huh.
the intronic regions as well, and just see what the differences are. Because I mean, the regulators are probably why the animals look different, and you wouldn't be getting the entire picture of it, but you'd be getting
Yeah.
a unique extra element to it. But again, price does come in. Even today, like the paper we're looking at and we're going to chat about, like that also I think had price come in.
Yeah. Gotcha. Yeah. Huh. Really interesting stuff. And again, I'd like to point out for the chat just how unique and cool an opportunity this is to be able to hear a geneticist talk about his craft, talk about his field of expertise, and be able to go into the weeds in a way that's still kind of understandable for somebody who doesn't have a background in this. Like, this is really, really special. So I hope you appreciate how incredibly cool this is, chat. Holy moly.
It brings me so much joy because I'm the same way too. As we're talking about the paleontology element to it, and then my favorite is when we both kind of have like a set idea behind the paper from our unique perspectives, and then the ideas flow together. Be like, okay, well, what can we do with these two disciplines to make a new question? Like those have been my favorite part of the crossover. Where it's like, wait a minute, what else can we can do that would be really cool?
That's why I was so excited about that paper, the living fossils paper, because I felt it was a perfect kind of dovetailing of our two different fields. Oh man, I'm excited for this. I kind of feel like we should discuss this first because I honestly don't know if we're gonna have time for the other one. I'm not under time constraint tonight either. Like I can go for a while, but I just feel like this is going to take a while to discuss, and I'm really excited for it.
I think we should focus on this one first. Yeah, I agree. We can always save the next one for next week. But
sure.
It's like you said, it's a really nice pulling together of both of our fields on this study because it's. I don't know. Would you mind introducing the idea from last week about how we had talked about the living fossil and you found a paper that you would point at and be like, this is not real, and then how we got this one.
Yeah. So here, let me grab the cycad off of the shelf. Here we go. This is a plastic version of a cycad. I would have a real one in my office, but there are cats who live here in the same house as me, and they would chew on it. Cycads are toxic, so I don't want them doing that. So here's a plastic one. But this type of organism is called a cycad. And so it is a plant that's often been called a living fossil. So these were kind of some of the dominant land plants during the age of the dinosaurs. They were everywhere, super diverse. It's a plant that might look like a fern or a palm or something like that, but it's neither. Its closest living relative is probably a ginkgo. And there are cycads that are around today, too. But there's this idea that, this is a living fossil, that these organisms are largely unchanged from the time of the dinosaurs. But the paper that we looked at last time was kind of calling that idea into question, like challenging the very concept of a living fossil. Because it turns out that the cycads that lived during the time of the dinosaurs are very different looking than the ones that live today. And they've seen a tremendous amount of diversification since the time of the dinosaurs. And, so that paper was kind of questioning whether the term living fossil is actually useful or instructive in any way. They're like, we should probably kind of abandon that term because living things are constantly changing and evolving. And, if you're not evolving, that's because you're extinct. You evolve or die because conditions are constantly changing. And even if conditions aren't changing that much, you still have stuff like genetic drift. And looking at this diagram here that you pulled up, Bullet, from the paper, there's so much disparity between different groups of cycads today that it's not like they're unchanged at all. They've changed in a bunch of crazy, weird ways. They all look so different from one another. They diversified tremendously. So it's That's saying that cycads could not truly be considered living fossils in this regard. I guess the term living fossil is meant to imply that there's a group of living things that existed a long, long time ago, tens of millions of years ago, but they also exist today, and they've changed very, very, very little to the point where they'd be, nearly identical to the ones that lived a long time ago. That's kind of what living fossil is supposed to mean. And as paleontologists, a lot of us are kind of, I don't know, living fossil to me has always been a term that's like, oh yeah, superficially that might look similar, but I'm sure they've changed a lot. But this paper that we're going to discuss right now says that no, some organisms really haven't changed hardly at all. And I'm really excited to get your perspective on this book.
Yeah, so Lita found this paper too, and we talked a little bit about it last Friday on Science News, and we both were like, Lita, I was telling Lita about this paper, and we are like, but we got to talk to Danny about this, because this is like,
yeah,
it's such a unique and very different flavor perspective, where it's very easy, I think, like the argument that was in the paper from last week about everything structurally looking different makes a lot of sense as to why they're not a living fossil, because they're different, right? You looked at that graph and all of the plants look different. Great. But this one, right, is doing actual sequencing. So they sequenced over 1,000 exons. So that's, I think, important to note here, is they were looking for protein coding regions that are exons.
Gotcha.
So they, like that intronic...
Those parts of the genome are where the rubber meets the road, figuratively. It's like, that's what actually contributes to the structure of the organism in a direct and material way, right? Or is that oversimplifying?
It's hard, right? Because the introns are those regulatory regions.
Okay.
And so they've ignored the regulatory regions and just took out what makes the proteins.
Okay, gotcha.
And so there's still some visible differences in these animals, right? And we know it's not from the protein coding from this paper. It's from those. It might be from the regulatory regions.
Okay.
So I think those are important, but they didn't look at them in this paper. And is it because... Okay, but the cynic in me... the cynic in me says this is the journal Evolution. Which is a top-of-the-line evolution journal, but in the global scheme of where it falls...
It's not a genetics journal.
Well, no. It's mid-tier. It's mid to low tier in terms of impact factor.
Oh. I see what you mean.
And so I think money and the aim. So I don't think that it's not they were good geneticists. I think it's just the limitation of working on a non-model organism.
Money.
As Tommy mentioned, triggered the money command. Honestly, I think that's what it is. That because you're working on fish that are not yet known to be,
Yeah, there's no like biomedical application for them or something. They're not, I don't know, and they're not Xenopus African clawed frogs or something that actually have like.
And it's not that they don't necessarily have something that could be applicable to biomedical use. It's just that really short-sighted look of like, well, I don't see it now, and therefore, why would we throw money at something like this? Which is, a very short-sighted, I think, way of looking at things. And Hanny asks...
I don't know. Yeah. Hey, sorry, go ahead.
No, you go ahead.
Well, I was going to say that, garfish, the Lepisosteans and Atracosteans, these are... If we have any, fishermen in chat, if you're familiar with gar, you probably know them as, a garbage fish. So even in, fishing circles, fish and game, they're not considered to be, a high esteem kind of clade. They're... So, I don't know. Gar don't get nearly enough appreciation. And even as a paleontologist, I can tell you stories about, oh, shoot, another gar scale that I just found. So it's nice to see them get some love in this paper.
By scale, do you mean, a physical scale or, a genetic
physical scale?
Okay. So I was like, wait a minute, what kind of scale?
So each of these animals, they've got these lovely ganoid scales, so it's like a bony scale with covered with ganoine, this really cool substance that they have. It's almost like chainmail armor.
Okay.
These fishes are, their scales are little bits of, of armor. It's like, was it Lord of the Rings? They had dragon scale armor or something like that.
Probably, yeah.
This is a real animal that basically has that.
Cool.
Which is really, really
and
cool.
Does that mean it fossilizes any easier?
Oh yeah.
Okay.
Really, really easily. And each of these animals has hundreds or thousands of these ganoid scales. And so when the animal would die, they would just go all over the place. You end up with thousands of them. All over the place. And so they're one of the most common fossils that we find in, say, the Hell Creek Formation or the Judith River Formation. If you're out looking for T-Rex or Triceratops in the Hell Creek, you're going to find 100 gar scales before you find, a single T-Rex bone or something like that,
Which is a great segue, y'all. There is this Amazon link right here that you can help support Danny on his fossil excursion.
We're gonna dig up some gar fossils hopefully in Wyoming this summer with the use of this equipment. Yes.
For some reason, I can't pin the message right now. I'm getting like an error thrown, but I'm gonna keep copy and pasting it, y'all. This is an easy way to support Danny on his mission and being able to bring you live fossil science from the field. I actually opened it up today, Danny, and I was like, "There's so much that had already been gotten off the list." I'm like, the community.
Isn't it crazy? Yeah. Ethan was texting me this morning. He's like, these items are just disappearing. Like the generosity is astonishing. We've got Mini Pie, who's decided to take center stage right here. But yeah, she loves being on camera. Mini Pie.
Today I thought that the camera had just died because right her color she stepped in front. I was like, wait a minute.
Yep. She's got a tendency to do that for sure. And by the way, if anybody purchases any items off of that wish list, Ethan and his girlfriend, my colleague in the field and his girlfriend, they have a cat too, and he's been sending me all these photos of the Amazon boxes they've cut holes into, and like their cat Java's peeking out of the holes, and she just loves those boxes. So not only will you make a bunch of paleontologists very happy, but also you'll make a cat very happy too with any purchases that people make off of that wish list.
Perfect. We've been plugging the wish list, Danny. So I'm glad to hear that.
I really appreciate that, Belinda. I really do.
It's a little thing.
That stuff will be. Sorry, go ahead.
It's a little thing that we can do to support.
I appreciate it a lot. A lot of those items are also, they'll last a really long time. Like, there will be future generations of paleontologists who will still be using those picks and shovels and chisels and everything, for decades into the future. So, it's one way to make a tangible material contribution to the progress of science. So,
and as well as because like the funding is so limited, this stuff isn't cheap, and so it's a great way to offset some of the costs for supplies by supporting this way, and then the money can go towards other, being in the field and looking for the fossils and making a big change and contribution to society.
Definitely, yeah.
Sorry, Dan, I had to get the plug in. You know this.
I appreciate it very much, Glenn. I really do. It means a lot. I appreciate that very much.
Hanny had the question earlier if you could track the junk gene evolution. So, Hanny, if they had unlimited money in this data set, they could have done extra sequencing, pulled out intronic regions or these regulatory regions, and looked at differences as well. And they have like these really nice graphs that are common in these kinds of papers, where you're looking at differences in exon between different species. So, for example, we're going to ignore what this actually is right now, but just look at the colors. If you're looking at a particular exon spot between two species, it would be, like this nucleotide is pink, this one's pink, that's a match. And then there's another match, and another match, and another match, and another match. So you could look at that across different species and see where there's matches or where there's gaps or where there's changes. And so you could definitely do it. Now, it's just harder, Hanny, to figure out what it means because it's actually easier to mutate a protein coding gene, because if you get rid of the protein, there's a measurable difference. You can look in this animal and be like, oh, it's missing this protein. Let's see what the behavior is like. Versus if it's a regulatory element, it could be early in development, it could be later in life. You never know exactly when it's going to become important. So function is much harder to show in changing regulatory regions. But yeah, you can absolutely do the evolution of them. All right, so yeah, this was the garfish. I think they look really cool, Danny. I don't know how you feel about them.
Just to show the chat, they are. Some people may have heard of alligator gar before. That's just one species. I think there's like 6 or 7 living species.
Yeah.
But they are really, really cool fish. Minnie, are you knocking stuff down? Knocking the lights off my desk. Be careful with those, Minnie Pop. Yeah. They are just some of my favorite extant fishes, because I've dug up fossils of these, and the fossil ones look remarkably similar to the modern ones. It's kind of astonishing just how incredibly similar they look. They're really, really wild. And so right there you've got...
Yeah, I pulled up just to see what they look like swimming around.
And that's a fairly young one. The big ones, these critters get up to like over 3 meters, like well over 10 feet long.
Yeah. Our friend,
huge.
We do have a fishing friend in the community, Tatum. He said he's caught them in the past too. He's never eaten one, but because actually how you said there's the idea of the garbage fish. But he was saying it was like a 10-footer or so. He didn't send it publicly. He sent it privately of him showing holding one that they released back into the water. But it was just like this beast of a fish.
It's crazy. Like, they've got really, really big ones at the California Academy of Sciences here in San Francisco. And some of them are like over 6 feet long. And they just have these giant, gnarly, alligator-like jaws. Like, they're genuinely kind of scary to look at, even though I don't think they've ever really harmed a person.
So that's been shown, yeah. Like they haven't. They're apparent. Like there's no evidence of. Never say never, right? But of there being injuries from these animals. But like their teeth, it's really imposing to see like how it's coming out of the skeleton.
Yep. Their teeth are really nasty looking. They're so cool. I think this is maybe a needle nose gar here, or maybe a spotted gar in the video.
Yeah, this one's showing a freshwater gars.
They're all freshwater, yeah. There's no marine ones that I know of. But they tropical gar, yeah, there you go. Very nice.
Raiders, welcome the heck in, Hazel. Hazel, how's it going? How was the stream? Tell us all the things. Let me see if I can spell as well with the shout-outs. Hazel, welcome the heck in, Raiders. Oh, you're playing some Minecraft. Nice. Hazel, tell us about the Minecraft. Did y'all build anything? Was it scary? Were there things coming after you?
We can rule the galaxy.
Oh my goodness, and hi! A raid and a resub? We're not worthy. That is...
Hazel Azazel just subscribed for 3 months.
Hazel, thank you so much.
Also this,
Hazel, thank you very much for that resub. That's a piece of raw science. And a partner plus point. Thank you so much, Hazel. Welcome the heck in, Raiders. How was your stream? Tell us all the details. My name is Balint. 1/3 of Science streams. The other 2/3 are my lovely wife, Lita, and our daughter, baby, Alona. Lita and I are research scientists with our PhDs in molecular biology, and we do live science content here 7 days a week. Today, we're joined by Professor Danny Anduza right over there, single greatest science communicator on the planet, and paleontologist, real-life paleontologist. You can hit the follow button underneath in the episode description. And we're talking a really cool paper looking at fossil science and genetics and challenging the idea of what a living fossil is today. So y'all, welcome in. Hazel says, I was working on a fully zoomed out 4x4 map of my solo world, all peacefully made. No, listen, that is not being a baby. I prefer peaceful mode and no enemies. I also don't play Minecraft because I would get hooked on it. But I would 100% see, like, being able to build stuff, not having to worry about people breaking it up, and just having a nice time. Hazel, that sounds lovely. Thank you so much for the raid and for that resub coming in. If there's anything you want to drop, any socials for the chat, please feel free to share it. Welcome. We also have Kelly Cakes in his house. Kelly, how are you tonight? Welcome the heck in. Naderfiend, thank you for the 100 biddies. Kelly said earlier that in Minnesota they also had long-nosed gar where she was growing up.
Yeah. They still do. Yeah. They're really, really cool. They go all the way up through. I want to say we have them in Montana, or I used to live in Montana. I want to say they go up there. I know they've got sturgeon and paddlefish up in Montana. I want to say they have gar too, although I found a bunch of gar fossils from 66 million years ago in Montana, so maybe that's why I'm thinking that. But yeah.
Alex, and speaking of armor, Alex says the alligator gar scales are inspired material for safety gloves. That's really, like you were saying, also like the chainmail armor. Yeah.
Yep. Because I think if like if somebody goes out fishing and catches an alligator gar or any other kind of gar, especially a big one, they've got a lot of difficulty in like cutting them up if they want to eat them. So I think they need to use like a hacksaw or something like that in order to actually like cut through their flesh, because you've got that crazy ganoid scale armor on there. Yeah, it's really wild. They're naturally armored fishes. Super cool.
Hazel debuted the new channel mascot, Danny. And so, let me scoot it around a little bit. There is the plushie version. It's a pangolin, pangofly. Half fly, half pangolin.
Oh, that's kind of. It almost looks like it has some axolotl in there too.
Oh yeah, the ears. I had to show it because I know how much you love World Axolotl Day as well, and like celebrating and preserving the axolotl. I'm like, oh, that's really cool.
Very cool. Yeah.
Very cool. Hazel, thank you for sharing with us. Welcome on in. And again, Raiders, great to have y'all here. If y'all like science, stick around. Hanny actually, or not Hanny, we had Naderfiend asked a question about, is it possible for species to not evolve? Naderfiend, you're in the right place because that's the paper we're talking about today.
Yeah, I would have said no, absolutely no, but then I read this paper today, and they're still evolving, but it might be very, very slowly compared to what I would think is typical. Yeah.
Yeah, these are the different species that we were looking at here, and for the Raiders coming in, we had talked about this concept of a living fossil last week, and the idea being that it's, we were looking at different plants, and they were, they looked very structurally different, not even close, and it was like, all right, I very easily can buy the idea that a living fossil is like a buzzword that you put into a grant, and it's just something that gets people excited, but not necessarily a real thing. But this week, we're talking about one where they looked at the genetics, and the question was, so chat, they looked at over 1,000 genes between these fish. How many of those genes do y'all think had changes that resulted in different amino acids? Meaning that, 3 nucleotide sequence to make an amino acid. How many of those 1,000 genes between these different animals had a mutation that yielded a different protein? Claire says less than 10%. 10 says Jim of those 1,000. 3 says Alex Vixen. The correct answer is 0.
That is nuts. That is wild. Holy cow.
And I was blown away hearing that was the... And this is, y'all, like, the common ancestor of these animals was, like, over 100 million years ago.
Yeah, it was back in the Jurassic, I think.
Yeah.
Or early Cretaceous, maybe? Nuts.
Yeah, early Cretaceous, yeah, for the split
Early
point
Cretaceous
of the
makes
groups
sense,
that they were focusing on, yeah. And it's wild to me because, a couple of caveats, right? They looked at a little over 1,000 genes. They only looked at exons, which are the protein-producing regions. They didn't look at introns, which are the regulatory ones. So there may very well have been changes in the regulatory elements, or outside of those 1,000 genes. But the reason they looked at those genes is because they were actually being able to identify what is a function of that gene using homology. So Danny, you asked, is there a database of these genomes? Where would I go?
Yeah.
So you can go to PubMed and you can, what is called BLAST. You can compare a genome that you sequence to existing ones, and if they've curated what the gene is, it'll fall into place. Like this part of your genome that you sequenced, these genes match very closely to these already described genes, and so we kind of can predict what their function might be. You have no way of confirming it, but it's a good prediction because you've confirmed the gene in another animal and it looks very similar. And so they wanted to look at something where they actually had here is actual data on what this gene is and what it might be doing.
Gotcha.
And so again, I don't know if they went outside this 1,000 genes. If they did 2,000, would it be different? Is it still 0? They didn't really go into I don't want to say cherry picked, right? But how exactly we got to this kind of number is very surprising to me.
Right.
And so that to me leads to whoa, well, why? Why is that? What about that 1,000 is special? But the fact that it is 1,000 is remarkable. That there's no changes.
Yeah.
And it's the same proteins.
And to me, this is... I don't know. I think my underlying assumption as a paleontologist has always been people call GAR living fossils, but that's just because we don't recognize how much they've changed over the past 70 million years or whatever. There's always this underlying assumption for me that genetically, via genetic drift or other processes like that, or even just selective evolution, they must have changed a tremendous amount in terms of their genome, even if their phenotype hasn't changed all that much. And I kind of assumed that maybe people who study GAR, people like David Solomon is a pretty well-known GAR researcher. He's got to know all of the differences between, they've got to look night and day different, these Cretaceous GAR that we have as fossils versus these modern GAR that we have today. I'd always kind of assumed that they must have changed a tremendous amount, but I don't know anything about GAR, so I'm not going to see those changes. But what this paper is arguing is that no, they just haven't changed that much. Genotypically, and that's a revelation to me. It's very, very surprising, borderline shocking, I would say.
And it's not just 30 million years ago, right? That this break point happened where the change is, 30 million is still huge between us, right? But it's over 100 million years.
Yeah.
That over 1,000 genes have had no change.
Which is wild. I mean, to me, it raises the question, why is this particular group not subject to very much change, whereas you have teleost fish, fishes with those movable mouth parts and stuff like bass and trout and salmon and everything else. Why have they changed so much while GAR have not? Is there something about the architecture of their genome that allows them to change that much that these guys just don't have? Or is it a lifestyle difference? Is it because these guys, because of their spawning patterns, they don't get allopatrically separated, so you don't have allopatric speciation that occurs? What is it about? What is the mechanism that keeps them from changing that much? And that part kind of sailed over my head, Belit, because I'm not a geneticist and I don't understand a lot of the terms of art that you use in genetics. Do you feel like there was a strong argument in there? Did that make sense to you as a geneticist, why these animals are not changing that much?
I don't think they proved why they didn't change much.
Gotcha. Okay.
To me, is my takeaway from it. I think, but real quick, because I'm going to rabbit hole to the last figure. But Hanny was wondering, what kind of changes that are non-physical might be happening. As Danny said, if they're separated geographically, there could be reproductive changes that just like, for example, Hanny, chromosome number in fruit flies. More distant species have more chromosomes than modern, like the newest flies, like melanogaster, because they have chromosomal fusions. And so you can't mate those two animals anymore, not because their reproductive bits don't interact, but because genetically the chromosomes could no longer pair up because you have a fused chromosome where one chromosome used to be two in the other animal. So that's like a non-visible change that would make these animals very different. And so, like, I think the coolest part of the experiment, like, you know, I think it's amazing that we're seeing this, like, 1,000 genes that aren't the same, or that are the exact same between these animals that we think are different. But then, y'all, you know, on our stream and Danny's stream, we always talk about the concept of what is a species, right? And it's
species concepts.
It always. But, like, would you say, Danny, that the end-all argument is if they can't interbreed, then truly they are different? Like, if they can interbreed, if they can't interbreed, or if they can, the hybrid is non-fertile, then those are truly two separate species.
I can't say that as a paleontologist because I can't falsify that for fossils. So I can't say that. Yeah, we've got to have a different species concept as paleontologists, you know?
And so that's the cool part here, I think, is because our definitions are coming together. So in the molecular level, if you can make a genetic mule, right, where you mate two different animals, you pull them together, right, they can produce a viable offspring, but that offspring is no longer fertile, then you've got a dead end, right, and they are in fact two different species. Tommy, sleep well, Tommy Platypus. Here, they took two species of gar,
two genera, I think.
Yeah. Different parts of the tree.
Patricosteus?
Yeah. Here's the Patricosteus, and then which one was it? Was it the simplex?
Was it Lepisosteus and Atracosteus, or did I get that mixed up? Atracosteus, I think, is the alligator gar. Lepisosteus is like spotted gar or something.
Oh, it's Lepisosteus osseus and Atracosteus spatula.
Atracosteus spatula. So Atracosteus spatula is the alligator gar, if I remember correctly.
Spatula.
So I think they bred that with a needle nose gar. Is that it?
The osseous one. With osseous,
nuts.
This one was one parent, and then the bottom one was the second parent.
Yeah.
And to me, it's crazy because the offspring looked like a mix. So the middle one here, chat, this skull is the offspring of this inbreeding, and it looks kind of like an intermediate phenotype. And I think the reason they picked it was because if you look at the skulls, they're so different. Like, it's that needle skull, right? For one,
long nose gar is what they're called. They're
long nose,
not needle nose. That's the long nose gar. Long nose gar and alligator gar. They should not be able to interbreed. That's insane.
Yeah, especially like if you're talking 100 million years of evolutionary split time. It's like, okay, sure, they made an offspring. Okay, that's fine. But then this offspring is still fertile. Right? And so that, I think, further adds to that they aren't... That the idea of living fossil, I think, comes back, right? Because they can interbreed, and those offspring are viable with no fitness detriment that was at least measured. And so, like, what is going on there?
It really messes with our idea of species concepts, for one, but also, like, if these critters have been separate for tens of millions of years, you should have enough genetic change that's stacked up in that length of time that should prevent them from having viable offspring. Wild. Just nuts.
And the neat part too is, at least in the genetics that they looked at, like, there can be interbreeding, like Alex mentioned too. Like, there could be a dolphin and killer whale hybrids. There could be fertility, and the same thing with some fruit flies too. Like there is some fertility there, but they're sickly, or like something's a little bit off with them. There's like, or like the lifespan's shortened. Like
there's a serious like fitness deficit there for sure.
Yeah, but it seems here, at least in the genes they looked at, like it's very similar genetic components. Its genetic elements are maintained and the same, and the fish seem totally fine. Like they grew this animal up to adulthood to get the skull.
Yeah.
And there was
and
nothing.
Look at how beautiful that is. It's just like a kind of a perfect cross of those two right there.
It's hauntingly good. Like I imagine
I'm
like
a little bit weirded out.
A paleo honestly. Artist, if you just told them like make the hybrid, like that's the easiest thing to draw, right? Okay, intermediate snout. Here are the same bone lines. Okay, good. It's like it's eerily on point.
It really is. Yeah.
And so I guess just the foundation, like Danny, what do you think of the species concept? Like, how would you like if you were just looking at the fossils? How would you think of these if you didn't know that this was a hybrid? If you were just looking at these skulls, from 100 million years ago, one might predict right that they're different species. No,
if I had these three right here. If it was just the top one and the bottom one, it'd be like, okay, these are probably different species. They're around the same size. I don't know if these are to scale. They're probably not. Well, I know long-nosed gar don't get nearly as big as alligator gar. But it'd be like, okay, yeah, these are different species. But if you have an intermediate like that, and they're all from the same horizon, geologically, it'd be like, okay, well, this is an ontogenetic sequence. It's like, this is just one species growing up over time, like...
It's almost like the mini T-Rex.
Yeah, Nano Tyrannus. Yeah.
Where it's like you start to not want to name them as different ones because it could be, like you said, even intermediates.
The one that looks small and young could just be because it's small and young. It might not be a different species. But yeah, I don't know. This is wild. I wish these were to scale because I feel like that would kind of help me understand this a little bit. I feel like they scaled them all the same just to, to make a nice figure. But,
let me see if there's anything in the figure legend about scale, which, it would be nice if there was. Nope, it's just phenotypic intermediacy in the punitive skulls.
Right, yeah.
And then, let's see... Let me take a quick look at the supplement. Maybe they pulled something in the supplementary information. Okay, yeah. I don't know. I feel like that should be like you need scale bars, right?
Yeah. Yeah, and actually there might be an indication there where no, I don't know. That probably has something to do with how they prepared the specimens. All
right, that's the supplementary table. And see, Chad, this is the cool part too about these crossovers is that we notice different details that the others like I don't know why they would necessarily need that, and be like, no, I think we do need this one, and it's cool to have that level of perception.
Definitely.
Let's see, I'm just cycling through all the supplement.
And it was, to me, it was kind of wild too that they did like geometric morphometrics in this as well, because that's something that to have that mixed with a study of the genome was pretty cool. I know some people at Berkeley who do this kind of work on trout, and to see this done on gar is really, really neat.
Yeah, and super surprising as well, because again, it's like a non-model system. So just being able to see something like this being worked on. No, I think
seeing
it's really
gar
cool.
get some love, and not just zebra danio or what would be another? What are some model organisms for little fishes?
Danio is the biggest one, honestly.
Danio, yeah. Yeah.
And that's
no relation to me, Chad.
I wasn't gonna say it. But I mean, it's because there's so many tools that have already been developed. It's kind of like you just fall into the trap of we have tools for this, and so we're just gonna stick working on this for now. just because
I wonder
it's
if
easier.
this will prompt some work on Gar in the future. That would be really nice to see Gar get some more love in this regard.
Yeah.
I feel like I need to do a quick shout out too for Gar researcher Solomon R. David is a PhD in aquatic ecology, and he's got a big social media presence. He posts on Twitter all the time about Gar, and probably Instagram and other platforms too.
Nice.
I don't know, I'm not that. But he's always making all kinds of Gar puns and everything. But he's like the Gar expert. He is the Gar guy who gets interviewed. He's always trying to advocate for the conservation of Gar and their appreciation as a group of animals. And he's probably pretty excited about this paper, I would imagine. I'm going to see if I can interview him at some point on my channel, because I've got a long history with Gar. But
Oh,
yeah,
Dr. Abson
yeah.
says that he's a co-author on this paper too.
Oh, is he? Oh, shoot. How did I overlook that? Darn. But yeah, he's a cool guy. I'll put a link to his Twitter here in the chat if anybody wants to follow him. But yeah.
Oh, look at the picture too.
Yep. Yeah, that looks like a
Terrifying.
young alligator guy right there.
It's
Atrocostes
terrifying.
spatula.
Oh,
Yeah,
Real Men Grow Beard says the long-nosed gar skull can be 7 to 15 inches. The alligator's gar skull is 15 inches.
Well, they can get bigger than that too. Beards. Alligator gar can get truly enormous. Like they can have a skull that's like, I want to say 2 or 3 feet long. But yeah, they are pretty cool. Pretty cool animals.
Oh, and I think they're beautiful. Probably not the most popular opinion, but
I think they are too. I mean, So, remember that game Saurian?
yeah,
That I've played a few times.
yeah.
So there are gar in that game.
Oh, cool.
'Cause they lived in the Hell Creek environment, and they're one of the critters that they actually render in that game. They swimming around. You can catch them if you're good at it.
I really I love you playing that game because it's so cool to have the highlight of what you see in game versus what the reality is, and it's just a lot. It's always so much fun to watch you play that.
I'll try to do that again before I leave for the field this summer. We'll see.
Don't get me wrong, I love shredding Jurassic World too, but it's also it's cool to see
I definitely enjoy Saurian a lot more. So it's not. Yeah, it's a fun experience rather than a painful one. Yeah.
Yeah. So thank you, chat, for helping us on the size. I didn't see anything pop up in the figures. I'll go ahead and throw all the figures on our Discord. It is open access, so y'all shouldn't have any problem getting a hold of this paper, which is really nice because usually Evolution's behind a paywall, so they must have paid the premium price to get it.
Good for them.
But there was this a nice little workflow that they had as well that jumps through, each of the samples that they looked at, the phylogenetic sequences, and how they did all the matching and looked at different clades as well and did comparisons elsewhere in the paper. So at least it's a nice little workflow, because elsewhere in the supplement they also have comparisons for other animals that may have had that much evolutionary time that we have evidence of, and seeing what the spread is. And so it was just really highlighting that the spread for these gar is so much smaller than for other animals,
which is nuts. Yeah. Were you as surprised by this paper as I was?
I was shocked.
I mean,
I was blown off my about all of them, right? Because I think the evolution, the lack of change, because I'm like you too, right? If there's not a phenotypic change, like a physical change, I assume on the genetic level something's going on.
It's got to be some drift going on, right?
Yeah.
I mean, it's that's the shorthand that I always use when people ask me oh, Danny, which animals have not evolved for the past 100 million years? I said, well, no animals have not evolved. If you're not evolving, it's because you're dead. But this kind of runs counter to that, and could you maybe outline, as a geneticist, what is your understanding of genetic drift? Can you explain that for the chat? Because I feel like that would be useful.
Do you want to do a compare and contrast, like because you were using it as well, like what you think of it, and then what I think of it, and see how our definitions match up?
Yeah. Sure. Should I talk about my understanding of it first?
Yeah.
My very limited understanding of genetic drift, not being a geneticist. So, I remember back in university, my main experience with genetic drift was talking about it in a seminar class called Macroevolution and Fossil Record. And we were looking at a book, big old book by Stephen Jay Gould, right here, called The Structure of Evolutionary Theory. This doorstop right here. This is how many pages in this book? Let's see, 1,433 pages. Yeah, this is like Stephen Jay Gould's like last master work. But he kind of talks about the history of the study of evolution, and he kind of like tries to bring it up to the present, and it's just an extremely long book. The guy really needed an editor, and apparently did not have one. But anyway, he goes into, there's like probably hundreds of pages about this idea of genetic drift and the drunkard's walk, which is kind of the illustration that I remember when I think of genetic drift, where it's this idea that like genetic drift is basically like non-selected evolution. It's change in a genome over time, just kind of at random, where it's like, if evolution can be described as the change in gene frequency over time, like certain genes become more common in a population, it might be that like just based on random factors, certain genes become more common and populations can kind of like drift genetically in a certain direction. Like certain changes can occur. It's not like they're being heavily selected by predation or environmental change or anything. It's just like, even if these critters are living a gentle, simple, calm, untroubled life, and they're not being picked off left and right by predators or environmental havoc or whatever, over time, their genes will just kind of change. And one of the ideas was that of like the drunkard's walk, which is this analogy where certain things, like certain genotypes, might become more common over time, even if they're not being heavily selected, just because there's some sort of like constraint on some side. Like they'll picture like a guy who's had too much to drink and he's walking down a street, and there's like a wall of buildings on one side and there's the street. It's like, where is he going to end up? He's not going to end up inside the buildings. He's going to keep bumping up against the walls and the windows and everything. Eventually he'll end up over in the street, just because it's kind of how things work. So if there's some sort of genetic constraint somewhere, you'll keep bumping up against that, but eventually, even at random, you'll just kind of veer in the other direction and you'll end up over there. And that was kind of like one of the constraints that I'd read about for genetic drift. Even if things are almost entirely random, you'll still get drift in certain directions because you don't have constraint, in one direction versus another. Does that make sense?
Yeah, and my take, well, the way I've learned it too, is similar on that front, in that there's like a big gene pool, right? And let's say there's some gene that is represented in a couple of different ways in this pool, and nothing's being selected for. It's just there's just these variants that are existing there, and then the question, chat, is like, you shouldn't see that same variant pool in your population over 100 million years, because there's going to be random things that happen that affect that pool. Anything from like there's two classic examples, right? One is the founder effect. So let's say that our GAR, like a group of them, like let's say with a purple gene. Yeah, exactly. Like there's a big wave. There's like some tsunami, and GAR, like purple gene GAR, end up in a different part of the world, right? Then they'll produce their own population. Those will be genetically distinct from the original location.
Yeah,
and so
they'll be more purple than the GAR in the parent population, because purple GAR just happened to end up landing in this lake or whatever, to use a silly example. Yeah.
I know it's a goofy one, but it's like it's illustrating the idea, of like, instead of having that large mix pool, you narrow it down because of some, like, it's not necessarily an evolutionary pressure, but just random chance. Or you can have the bottleneck effect, which, let's say, like an asteroid
like
hits.
cheetahs.
Yeah.
Or cheetahs are a good example. Shoot, where they had a big population bottleneck where like there were only a few that survived, and then so it's like the genes that they have now exist in all of our living cheetahs because it was just like a very small population that survived. Sorry, I didn't mean to interrupt you.
Or bananas.
Yeah.
Like that are near genetically identical, right? So there's
banana.
Yeah, there's no there's like the fungal protection or something that they can kill, and there's the lack of diversity now, and it wasn't necessarily because something was selecting for it. It just is a random chance happening. So like the fact that these animals, like, selection pressure could change the genome, right? Let's say a disease comes in and certain fish survive better because of this disease state, that would drive evolution to be acting on those genes. Or
That's
the
selective pressure right there,
yeah. Or the random chance, like different water patterns, right? And yet, neither one of those seems to be in effect with these animals because, again, those genes are non-changed for 100 million years.
Right.
So Danny, I have a lot of trouble wrapping this around my head because of, like, you were saying, like, there's those two very different kinds of, there's pressure and non-pressure, but both of them are going to influence the genome. And it's like they're not.
To me, I don't know. You've got this idea of stasis, which is kind of anathema to me based on my background in evolutionary biology and invertebrate paleontology. This idea of stasis, chat, is that, like, you've got a group of organisms that's not changing. They're staying in place, stasis, as opposed to, like, constant evolution. And there are a lot of paleontologists nowadays who subscribe to this idea of, like, punctuated equilibrium, or what George Gaylord Simpson would have called quantum evolution. This idea that, like, you've got long periods of stasis and then the environment changes.
Together, we can move a galaxy.
Oh, very nice, Kennedy.
Kennedy, thank you for that
reset. And then
reset.
suddenly, like, the animals have to have, like, rapid evolution right there.
Just subscribed for 15 months.
It almost seems like that's what's going on here. Where, like, you have a period of long stasis where these animals don't really have to change. If their environment is staying largely the same. If these, like, slow-moving fishes are just able to just cruise around and, eat whatever they can catch and just produce their offspring, and, they're just kind of lazily swimming around these rivers, then it almost seems like cartoonishly simple to me. Like, I want there to be more nuance or more, like, something more interesting or complicated going on, but it kind of fits this old... It almost feels like a Just So story that, like, oh yeah, well, these are very slow swimming fishes. They're just living in swamps. They got a simple life. They just eat anything that falls into the water and nothing troubles them. And, they just don't evolve very fast, But it really seems like that might be what's going on, which... Yeah.
What gets me is that, I looked at in undergrad, it was like the evolution of the immune system, and it was always this Red Queen hypothesis that you're always running
just to stay in place.
Exactly. Yeah. And yet, so, like, there's always these pressures, or even, like, right, they're living in water. Water temperatures are presumably changing, right? The mineral concentration's changing as, like, different pockets of water are popping up around the world from, like, the moving of continents or the prey of what they eat, right? Like, you would predict that there would be differences just because of something like that.
But I could come up with a Just So story in which these are fishes who are living in these big river systems, and it's like, well, the temperatures change, they just swim to a different part of the river. Or this part of the population just dies out, but they're like constantly part of this continuous gene flow because they're part of this river system that's all interconnected. So, like, they're not going to speciate very much because you don't have separation of these gene pools. If they're just part of this big continuous gene pool like this, and say the whole Mississippi River, which connects to the Missouri River, and they can just pass their genes all along this big long system, then it's not like they're going up alpine streams and ending up in these isolated mountain lakes and stuff. They're just kind of like, I don't know. They're simple fishes just living a slow, simple life.
There is an alternative hypothesis that I want to put forth that I think might be going on. They didn't really show evidence for it in the paper, but there is a way that they could do this. There have been shown other, quote, slow evolving animals that have really good DNA repair enzymes. So let's say there's still mutations happening in the exons, it's just that they have the repair machinery to fix it, and so you don't have the opportunity for there to be mutation because it's always fixed correctly.
You just have really low mutation rates because you just have really reliable DNA. Like, I don't know. It's like buying a Toyota versus a Jeep where it just doesn't break down that often.
Well, or if it does, right, the repair guy, he isn't like, your, national chain guy who doesn't care about your car. He's that mom and pop shop guy who takes a lot of pride in everything he does, and he goes and fixes it every time perfectly. And there have been studies where they've taken like the DNA repair enzyme from one animal and put it into like a human cell and seeing how well it's able to repair the DNA, and they've seen that, this guy is really, really good. Like this enzyme from a different animal that have low mutation rates. And so I wonder, I mean, they commented on that as a possibility in this, but they never pointed out the gene or actually done that next step of putting it into cells. Right. And Danny, it might not, it may very well be a combination, right? It might be that the lifestyle, and I think it has to be, right? Like the lifestyle is one where there isn't this active selection, and they've just been really lucky with the environmental niche they're in, where it's not like there's I forget what fish it is, but it's like one population in a lake in the Midwest that this fish exists now.
Oh, are you thinking of desert pupfish?
I think so, yeah, because
California?
Where
they only live in one tiny little like sinkhole environment. Out in Death Valley. Yeah.
And so it's not
they're
like
just totally isolated from all other fishes, and they
like it's surprising, right? Like with the recension of the icebergs and everything, like they just ended up in water bodies that
completely isolated.
Yeah.
So they became their own species, and like they have to be. There's a fence around the desert pupfish hole because, like, some no good nicks sometimes get into there, and there was a guy who he crashed his 4-wheeler into a desert pupfish hole, and they had to pull it out. But you don't have that problem with gar because they live in the whole Mississippi River Delta and up into the Missouri River as well, and a bunch of tributaries and lakes and everything, and they just kind of live all over the place, and so they're much less likely to speciate there, I guess. And maybe the hybridization thing comes into it as well, because it's like if there were another population that were to split off allopatrically, since these river basins, they used to flood all the time, then you would have the hybrid mix in back with the parent population and probably get outcompeted. So it's not like a hybrid or something like that, or you're not going to have that kind of isolation that leads to new species, I guess.
It's
Maybe
also,
that's kind of the idea.
Are you surprised, Danny, that the hybrid number isn't that high in these animals? Meaning, like they prove that there are these hybrids, right? But all of a sudden, actually, I do have the figure open. They're not just rampant. Like there doesn't seem to be this almost selection for the hybrid. Like they mentioned that there isn't like a fitness detriment, but there probably is something, right? Because we're not seeing the hybrids everywhere. So it's there's still, I guess,
Yeah.
probably some flavor of barrier in place, whether it be physical.
And I think, like, I can only think of a couple examples when a hybrid actually did really, really well in an environment and became its own species. We are actually an example. Like, certain kinds of people who've got, like, Neanderthal DNA, you could think of us as hybrids, I guess, in a certain extent. But also the Columbian mammoth is actually a hybrid species, apparently, between a woolly mammoth and an imperial mammoth. And they were able to survive, I think, because like a few imperial mammoths made it into North America across the Bering Land Bridge from Asia. And then they reproduced with woolly mammoths, and then they could just kind of take over because there's a big open niche there for them to exploit. And so they didn't have to compete against the parent species. I think that's kind of the idea. So it's like exploiting a new territory with different climate regimes and stuff kind of gave them the ability to outcompete the two parent populations and just become their own thing. Only have the most surface level understanding of Columbian mammoths and how they are hybrids or whatever, but that's what I remember, I think.
I guess it also brings into the question of what percentage do you need to be mixed of two different species to classify as a hybrid? Because I think, right, isn't the max that people have that we have today are like 5% Neanderthal right now?
Less than that for
Yeah.
Neanderthal. Yeah. So I don't know if that would qualify as a hybrid. You know,
It might. It's all like, again, comes back to this idea of what field you're in and the definitions that we have. Like one interesting feature, Chad, if you're at home now, if you feel on your head right about this region, if there's a divot, like a little indent like that runs across your skull, that is actually a Neanderthal feature.
It's clear as day on me. Holy cow.
Yeah, so that means you've got some Neanderthal in you. I've got the same thing. Lita doesn't have it. And so, and I told her, I'm like, there's this thing. And she's like, no, there's not. And she's like, oh my god, are you okay? Because it's like, there's this divot.
I think I remember it wasn't 23andMe, I think it was Ancestry.com that I did, but it said that I had it was in the 99th percentile for Neanderthal DNA or something like that.
Yep, there's a feature for you right there, that divot.
Oh, I kind of hate that it's like, it almost reminds me of like phrenology or something. Yeah. Ugh. That's wild. I'm gonna have to look that up now. Holy cow. But yeah, there it is, just
and Dr. Afson has it too, and they also have lots of Neanderthals. So we didn't sequence me, Doctor, but my dad did, and he's got the same thing. He's got that divot and a lot of the Neanderthal DNA.
Interesting.
And a first-time chatter, 3 green Legos. A cancer mutation might not be passed to the offspring of skin and organs battling ecological environmental stresses and mistakes of mitosis reproduction. So that's, I think, a really good point. That there could still have been mutation in these animals, right? It's just...
Sure, it's just weeded out, yeah.
Yeah, or it wasn't in the reproductive cells, right? So, like, they could be getting mutation in the exoskeleton, the skin, all the time, but the thing that's being passed on to the next generation surviving was very well protected.
Huh. And another thing that another chatter brought up, probably like half an hour ago at this point, was that they were wondering about predation pressures on these animals, and there's not too many critters that eat gar. They're not commonly eaten by birds, as I understand it. Alligators will eat alligator gar, and they'll eat other kinds of gar too, but it doesn't seem like as adults they've got very many natural predators. As fry, of course, they do, because they're born super, super small. But yeah, I wonder about that, and I wonder if slow development has something to do with that as well, because these could be very long-lived fishes. It's not like they grow really quickly. I think they've got kind of slower generational times, and that... I don't even remember that being brought up in the paper. Did they come up with that, Balint? Gestation periods? How long these animals take to reach maturity or anything like that?
No, there was no... And that's another thing, too, is because if you're older Like it's something in people
Uh-huh.
too, right? There's the increased likelihood that the embryos that you can fertilize will have some kind of developmental change, and that may or may not be a defect, right? Like resulting in a trisomy, or like where an embryo can't
Sure.
survive because it like one copy of chromosome 1 is not enough to survive. 3 copies will also not survive. You have to have 2 of, let's say, chromosome 1. But there can be transpositions as well. So there could be mixing of genes and even regions of chromatin where you do produce a viable offspring.
Yeah.
Right? And that would contribute to the population that could be undergoing genetic drift. Right? So even there must be, if I had to, as a geneticist, guess, I'd say there's something protecting the genome reliably, at least in the reproductive system, which is the most important, right? If you reach reproductive maturity and those aren't getting mutations and they're all like pretty stagnant, I mean, and if there's no selection pressures going on either, that's I think something really cool there.
What from a selection standpoint, in what kind of a regime would you have stasis be selected for, though? Like, in a situation like this, if there is some sort of a mechanism that's keeping these animals from mutating very much, under what kind of circumstances would that be advantageous? I guess if their environment isn't changing very much, then how would you have that kind of a mechanism selected for, I guess, is the question.
I see. If I was a gambling
maybe
man,
it just kind of happens. Maybe it just kind of happens, and that's like because you don't have a lot of change. You don't have a lot of change. Maybe I'm thinking about it in the wrong way.
I mean, I think there might still be variances in the population. It's just probably a really, really small number of them, like you were saying, and that really small number just never gains any upper hand because the majority of the population is this. Like, I wonder, actually, if we go back even further, let's go back 200 million years. Maybe that's what we need to look at. There was an evolutionary bottleneck. Maybe there was a selection pressure on these Gar ancestor at that point, and it was one flavor of the population that survived. And from that, like, genetic bottleneck where you're very limited on the genetic variants, radiated out everything. Maybe that was the method.
Interesting. Oh, I know that Gar first evolved in back in the Jurassic at some point. I want to say like Middle Jurassic.
Mm-hmm.
And then throughout the Cretaceous, they don't seem to change very much. In fact, they seem pretty similar to today. Throughout the Cretaceous, you've got the same genus, Lepus osteus, and the Hell Creek and in the Judith River formations, Gars that I've dug up before. And that's the same genus as today, like with your long-nosed Gar. I wonder if there was a bottleneck somewhere in like the Middle Cretaceous. I wonder if you could maybe even trace that back to like the Middle Cretaceous extinction event. The same event that wiped out the ichthyosaurs and the pliosaurs and the spinosaurs. That's an intriguing idea. I don't know how you'd go about testing it, but...
I don't know if you can, right? Because if... At least you can there are modern day survivors, right, that you can trace back and build this tree of relatedness. But I think you'd have to go even further back to get that kind of. So at least proving it in the way that they're proving here, I don't think we could. I think we'd have to go back to the other type of relatedness that you've told us, like about morphology, and seeing maybe if there's a I don't know how. If you can, in the fossil record, find evidence of a bottleneck, because if it's in the same strata, right, of like lots of different variances, and then maybe there was an event that made a bottleneck, if it's all in the same strata, would you see it?
Well, the way that we look at bottlenecks in the fossil record is just because you have a bunch of species that die out and one survives. And then things radiate out from there. That's how we identify bottlenecks. I don't know if it's as granular as you're talking about. As high detail.
So that would be like, if it were high detail, maybe one way that we could do that. But at least in how they're describing here with the genetics, I imagine we couldn't, right? Because again, they're looking at those exon differences. But I don't know, maybe that's...
We're trying to different scales here.
Yeah, like that's just the one hypothesis they didn't put forth, but maybe, like even this one in the Jurassic, this elegans species radiated from a bottleneck event. And maybe that's what caused it. And Charlie says if there's lots of variation, then 10 million years later, don't have that level of radiation, maybe that there was a bottleneck to it. Maybe, yeah, Charlie's. Golganak, sleep well. Thank you for hanging with us. So, I don't know, that's...
Yeah, I'll see you later, Golganak. Thank you.
It's a weird question. I say only weird because it's hard to know what exactly is going on, because we can't test that hypothesis, and I don't know about you, I don't feel any closer to resolving what's going on here.
I don't either. It kind of raises more questions than it answers, which is maybe the mark of a really good paper, but it's causing me to kind of rethink these things. Maybe we should look at that big diagram that they have with all of those different taxa from across vertebrata. They show like a Hoatzin and a coelacanth and a Tuatara and all of these critters, because there it is right there. Yeah. This to me was wild because it shows all of these like classic examples of living fossils, many of which I'm like really eager to discount. But can you interpret this figure for us, Belinda? What is the graph actually showing? Because I feel like part of this kind of sailed over my head.
Yeah, so they're looking at when there's an exon, right, that protein coding gene, it's in clusters of 3. So 3 nucleotides make a protein, or an amino acid, and then a string of those amino acids makes a protein. So they're looking for rates in which that amino acid comes out different. So of those 3 nucleotides, if you have a mutation, it'll pop out a different amino acid, which usually translates to a somewhat different protein. Whether it be different in function or in form, it might have the same function. It's just going to be structurally a little bit different. So, like, it's a way of measuring genetic differences, and it also accounts, like, if there is a synonymous mutation, meaning if it mutates but it yields the same protein, like the same amino acid, it does allow for this there to be evolution and effects of the nucleotides, but not, it just doesn't have any change effect on there. And so here it's showing the rates of that substitution against the divergence of the species. So what you're looking for is, if there's these wide panels, that means that there is a very small difference in terms of the divergence, or like the divergence itself might be long in terms of evolutionary time, but the exon rate of mutation is very low, and that's what.
So, like when we see the Gar silhouette there, when you've got a pretty long box there, that shows that these animals are not evolving quickly, I guess. Is that kind of the underlying idea?
So that there's... Yeah, exactly. If the numbers are closer together, that means there's a faster evolutionary pressure acting on them. And so, like, I think that's, like you said, I think this is, the coelacanth. I didn't even read it, but I recognize the structure of it, right?
Yeah.
That's the cool one to me, is because, like you've said, and we've talked in your stream a fair bit about this, is that... Hold on. The browser source won't let me zoom. Here we go. Come on now. That's the one that's classically called the living fossil. And it's pretty closely clustered together. Or other animals here, too. You know, I'd be interested in actually looking at, like, the Chinese sturgeon. They didn't really look closely at that one, but these are also box whisker plots. So it's taking into account all the dots or data points measured between these organisms. The bar is, I believe, the median, and then the box plot is getting your upper and lower range, and then the ones that are plotted above and below are your outliers relative to that average. So the 50% is within the bar, and then 25% above and below in terms of the variance. And so it's you're wanting to see if it's evolution's acting something closely clustered together, and yeah, we can look at these ones as well. These lower plots are looking at the mean percentage sequence divergence over millions of years, and so as you're seeing more divergence, you're seeing greater differences, right?
Gotcha.
Hi, Pen.
So the coelacanth right there, there's actually a significant amount of vicariance between the two different living species of coelacanth because the box is so much more squished.
Yeah.
But in the gar, since the box is taller, that shows that they're more similar.
Yeah. That the evolution's not acting on them as rapidly as you might predict. And this...
Gotcha.
So Gecko, basically wide bars are suggesting that evolution's not acting as a high selection pressure on these, versus closer together is there's more of some flavor of pressure acting on these animals, leading to be them to be more different from close relatives that are still alive today. And the coelacanth,
Gotcha.
right, is your class. I don't know how long I've heard that, is that it's a living fossil, like, forever. That one is actually pretty tightly compact, suggesting that the ones that are alive today, those two species, are pretty different.
Yeah, and that comports with some of my colleagues back at Montana State were working on a paper about that, where they sequenced the genome of coelacanths and looked at the differences and everything. And it turns out that they're pretty different from each other. The ones that live near Madagascar or the Comoros Islands and the ones that live in Indonesia, they're pretty different from each other, and that paper suggested that they are not indeed living fossils, because they've changed a lot. But that's interesting. Like I recognize a lot of other critters on here: Tuatara and Tinamou and Bishir and Queensland lungfish, and it's yeah.
Do you know why, Danny, they picked these particular animals? Like, I get the fish part, and like especially the lungfish, but like the tinamou and like these lizards, like why these animals?
Watson and that critter there on the right is not a lizard. That's a tuatara, which is this animal. It diverged before the first lizards evolved, so it looks very much like a lizard, but it's part of this ancient group called the rhynchocephalians. And they only today live in New Zealand. They're incredibly cool. They've got all kinds of weird features, like their teeth are totally different from lizard teeth. They're basically just like sharp points on the jawbone instead of like true socketed teeth that have roots and everything.
You've talked about these on your stream before. You've done deep dives on these. I remember that as soon as the image popped up, I remember this. Yeah.
May 2nd, we're going to be doing International Tuatara Day as well. Talking all about these critters.
Awesome.
Yeah, they're really, really, really cool. And they used to be much more widespread. They used to live all over the world in lots of different habitats. And there were so many different hundreds or thousands of species. Now there's just one left from this huge, ancient group. Used to be thousands of species. Now there's just one left. Sphenodon punctatus is the genus and species name. And this is a classic living fossil in the sense that it's the last one. So, like, we were talking about that last week with, like, one definition of a living fossil is like, oh, it's a critter where it's the last member of a group that used to be more widespread, more diverse. This is a classic example of that. And they look roughly similar to ones that lived during the Cretaceous and earlier. But I don't know. I always assumed that these critters were genetically very different from the ones that lived back then. But since there's only one species, what do you really test that against? With the gar, at least we've got multiple species, and you can see how different they are from each other. If there's just one species of these critters today, how would you deal with that as a geneticist, Blinn? I don't know.
That would be a tough one, because even if you're looking for the most closely related relative today, that might be a fair distance, and then you're...
really wide distance. Like, turtles and birds and crocodiles and lizards and snakes are all, like, equally distantly related from this animal. Yeah.
Yeah, that could be a very difficult one to test then, unless there's some kind of you've identified maybe a subspecies because of a different subpopulation that's maybe separated. Like there was a study we talked about last week about brown pandas. It turns out like brown and white pandas exist in one particular part of China in like this mountainous area.
Ailuropoda, like the giant panda?
Yeah. Like they are actually brown, and it's this one like mutation that's present in that population that makes them brown. They can interbreed with other, like there's no real, but you can maybe look at.
They're kind of in the process of speciating, maybe at the early stages of that. Like
yeah, like maybe possibly
years, maybe there'll be a new species.
Yeah. Yeah, and it was all down to this one pigment, and actually what they cloned the gene out from the panda, put it into a rat that normally is black, and as soon as it started expressing the panda gene, the rat turned brown, like after development, which was gnarly of a difference.
It's so neat and tidy and surprising. Like you would not think it would be that simple. Wow.
Yeah, and I mean obviously you can't test the genetics on a panda because.
Yeah.
The government owning it, plus also lifespan and ethics, like you get into a whole bunch. But they tested it on a rat, and they found like they put in this panda gene. This rat that's supposed to be normally black, came out brown.
Cool.
Yeah. But so maybe that would be like the test if there's multiple like subpopulations of them.
Multiple subspecies. Yeah.
Yeah.
Okay. Huh.
So, I agree with you. I think it's a really neat figure. Dearest Deer, thank you for the follow. Welcome in.
Nice. I was surprised that they also included the Hoatzin in this, too. Just to the left of the Tuatara.
Yeah.
Because this is like a classic example. I have this as an alert. Because the juveniles have got these claws on their wings that they use for climbing. And so they're this classic example of like, oh, it's just like Archaeopteryx. They've got claws on their wings and it looks like a dinosaur. But I don't know how how basal they really are. And it's that's an adult right there. The juveniles look way
even more different.
With claws on their wings. But
that's funny actually. The next search term suggested was Archaeopteryx.
Yep. And it's almost a cliche, and it's this example that we love to trot out as paleontologists when we're talking to the public. That some birds still have claws on their wings when they're juveniles, and it's just like the ancestral birds that evolved from theropod dinosaurs who had claws on their hands. But the idea that they're including it in this study is really interesting because I don't know. Those of us who were in the know realize that there are a lot of different birds whose juveniles have got claws on their wings, or even the adults do sometimes. It's just like an atavistic trait that sometimes they have. Birds like loons can have this. Ostriches have this. I think kiwi birds might have claws on their wings sometimes. It's like lots of different kinds of birds will have claws on their wings. But the Hoatzin is like the classic textbook example, and so it feels to me instinctually just like they chose this because Hoatzin, living fossil, but it kind of it seems like it actually checks out, like it actually works here.
Yeah.
I
guess like that was great to get like your take on why they picked these animals. Is that there's some flavor of it's almost like they're trying to prove the argument.
There's lore about them. They've got a reputation. They've got a rep, these animals. Yeah.
Or it's like there is such a thing as a living fossil, but it's not what you classically thought. Maybe that's also one element to it. Like we do have them, but you have to think about it in this, like kind of like how we talked about last week. It's not that like that plant group, right? They look different, and if you were to actually like just say they're living fossils, that's not enough. You need to do this extra rigorous amount to really prove, on an evolutionary time scale, that they in fact are some flavor of living fossil versus just, well, it's the last one, and so it must be, or it goes back a long time. Maybe that's why they had it like this.
That makes sense. And I'm looking at that plot right there. I mean, the box looks taller than it does in the Tuatara. And there is only one living species of Hoatzin. It's Epistocomus Hoatzin, I think, might be the genus and species.
Mm-hmm.
But, yeah, so I don't know what they're comparing it to. Maybe it's just, like, subpopulations or something.
Yeah.
But, yeah. And to the left of that, you've got the tinamou, which is considered, like, a primitive bird. Tinamous are, depending on who you ask, they either are or are not ratites. But tinamous are related to ostriches, emus, cassowaries, rheas, these big round-running birds. But tinamous are small. Sometimes they're called, like, people have called them Chilean partridges before. Even though they're nowhere closely related to partridges. They're part of this group of birds called the paleonaths. So it's tinamous, ostriches, cassowaries, rheas, emus, and I think the extinct moas, and maybe elephant birds too, are all paleonaths. So they're an early diverging group of birds. They split off before the neonath birds split off. And neonaths are 99.9% of all the birds you'd ever think of. But there are, I don't know, there's like 16 species of tinamou, or maybe more, or something like that. And it looks like their box is not very tall right there.
Yeah.
There's maybe a lot of variance between them, or... Yeah. Although they seem to be kind of on par with the Tuatara, from what I can see here.
Or like even like they look about the same as the lungfish, or even maybe a little bit wider than the coelacanth, but not by much. Sleep well, moonrise.
The lungfish I wonder about as well, because I don't know. Oh no, it's just Australian lungfish there. Shoot, which is one species. That's Neoceratodus forsteri is the Australian lungfish. That's just one species, and yet they're as opposed to African lungfish, there are multiple species of African lungfish. Polypterus is the genus, and I think there's like 7 species or something within genus Polypterus.
I wonder if there's just a baseline variance that you'd expect for within a given species, and that's what's one part of it's being plotted. But you're right, like if you're not specifying which of the African lungfish that you have here, it's a little bit confusing because, I mean, I guess maybe it makes sense because there does seem to be almost a subpopulation right up here. They don't indicate if it's a different species or not, but I wonder if if this cluster here is being represented by one of those additional species.
That's really weird. Yeah, I don't know.
And it's not in any of the other panels where it's like greatly separated out, right? And there's like an above and below. It's still a nice choice of species, I think.
Definitely. And this is like a classic smorgasbord of quote-unquote living fossils. I mean, they definitely did a good job of that. Because they've got, like Holostean, or, what is that? Looks like a chimera. The thing on the far left. What is that labeled? Let me look. Elephant fish?
Elephant shark.
Elephant shark, yeah. So these are chimeras, or ratfishes, as they're sometimes called. Which are, I've often heard them called living fossils. And yeah. Well, do you remember that, looking at the sturgeon and the Chinese sturgeon and American sturgeon there, do you remember that thing with the sturtlefish? The paddlefish sturgeon hybrid?
Yes, why? You must have talked about it on stream, right?
Yeah, I think there was a SciShow episode about it.
I think that sounds familiar, yeah.
Those are two groups of fishes, the paddlefish and the sturgeon, that diverged like 250 million years ago, or maybe longer than that. I want to say it was like in the early Triassic or something like that. It's like there is no way in the world that those two fishes that are separated by 200 plus million years of evolution, there's no way in the world they should be able to produce offspring, let alone like viable hybrid offspring. And yet there was a group in Russia where they kind of like accidentally did this. I think they just like left them in the same tank because they thought like, oh yeah, they're not going to reproduce. They did.
My goodness. Welcome in, Raiders. Oh my goodness, Suno Chicano. Suno, hello. Mew the Catboy. Suno. Edie Fanatic Akai, welcome on in. Y'all, how was the stream, Suno? Tell us all the things. Suno Chicano, y'all, is one of our friendos. He's a bard. He sings on his stream. He plays games. He does a lot of charity events, too. He's a gem of a human. Go drop him a follow right now. Suno, how was Kingdom Hearts at 1.5, 2.5? Was Feline Fancy guiding you tonight again to victory? Raiders, welcome the heck in. We talked a lot of lore of Kingdom Hearts. Nice. The Kingdom Hearts games are a lot of fun because I'm a sucker for the Disney universe, and it's a good time. Raiders, welcome in. My name is Belenz, 1/3 of Science Streams. Other 2/3 are my lovely wife Lita and our daughter baby Alona. Lita and I have our PhDs in molecular biology, and we do live science content here 7 days a week on Twitch. We're joined today by Professor Danny Anduza of Paleontologizing. You should know him by this point as the single greatest science communicator on the internet. You can follow him with the name highlighted in the channel description, and we're talking about this really cool paper that just came out on living fossils. We had thought we wrapped this up last week with a different paper that living fossil is an outdated term. It's silly, like it's used as a buzzword, and then here comes this paper of like, let's do some sequencing, and let's show that over the course of 75 million years, here's 1,000 genes that we looked at. And Raiders, guess how many differences there were in those 1,000 genes that they looked at between two animals, two fish that were distant by 75 million years of evolution?
It's gotta be like 900, right? 900 genes?
Zero. Zero of that 1,000. And they could interbreed after 75 million years of evolutionary split.
That's nuts. They should not be able to do that. They're breaking the rules.
And their offspring are fertile, so it's even more of a rule break.
That is so cool. This is so cool. Yeah. Oh, man.
But yeah, Raiders, welcome in. Suno, if there's anything you'd like to share, you know the drill. Drop it in the chat. Show it off to the party people. Welcome on in, everyone. We were just talking about the mix between the American sturgeon?
I think it was a Russian sturgeon. It might even been like a beluga sturgeon and an American paddlefish, because the Chinese paddlefish is now thought to be extinct. So it had to have been an American paddlefish. Yeah.
It's this cursed-looking fish, right, Danny?
The sturtlefish. Yeah. Where it looks like just like a perfect mix of a paddlefish and a sturgeon, which are wildly different looking fishes. I mean, they're so different from each other. The fact that they could still produce offspring after like 180 million years is nuts.
I wonder then if they did this same test that was described in this paper on those two fish, and like looking at those axons, would you get a similar answer?
I don't know. Yeah, and I was kind of surprised that they didn't include paddlefish in this, right? I don't think I think genus Polyodon is a American paddlefish.
Yeah.
I don't see that on here. I certainly don't see the paddlefish silhouette. Yeah. There was the video right there.
Yeah, here's that SciShow link for. Thank you, Alex. I'll throw it into the chat. Mother Nature, when I made the garfish, I made them right.
I guess so.
I think it'd be
don't fix it, you know?
a neat follow-up, interesting paper is just to sequence those two genomes and just do the same analysis on the exons, and
that would be really cool. Yeah, shoot. Wild stuff. Yeah.
Yeah, really exciting paper, I think. I guess, Danny, are we any closer to saying and coming up with the idea if we are how we feel about the term living fossil? Because I'm no longer set.
I used to be ready to just discard that term and say, oh, living fossil, it's a nice buzzword, but it doesn't really explain much. But I don't know what to think now. Yeah, which is exciting. You know, it's just really cool. And I feel like this is the sort of paper that I'm going to be talking about with my colleagues this summer when I'm out in the field. Um, yeah, I feel like this is going to come up a lot in conversation over the next 6 months.
It's gonna be really cool to see you find some more sturgeon fish, or not, and the
Yeah.
gars, or sorry, the gars and like the scales now, because like
Yeah.
I don't know, I've gotten a bigger appreciation for them from this
Uh-huh.
paper, because like they were neat fish, but like seeing these differences or lack of thereof.
Yep.
I mean, I don't know. You and I were hypothesizing about why there aren't differences for like 45 minutes and like bottlenecks and
Yep.
why there aren't there. There's just and I don't think we came up with an answer.
And I also think on a like science outreach level, as a paleontologist, like this is actually really exciting in that I can tell people like, oh yeah, Gar, you know, they really are largely unchanged since the Cretaceous. I can actually say that with my chest now, where I wouldn't have been able to say that before. There was always some reservation, like, oh, I'm sure there was a lot of genotypic changes. That you know, it's mostly superficial, the similarities, but no, it really seems like the nuts and bolts of these creatures are actually nearly identical to what they would have been back in the Cretaceous, and that is incredibly exciting. So I don't know. I'm jazzed about this, Belinda. This is
Yeah.
really really cool stuff.
Me
Yeah.
too. And thank you so much for letting us talk about this. Like again, it was just seeing this paper. I was like, it's the perfect paper for us to do a crossover on. I think it's the paleontology and the genetics,
Yep.
and I don't know. I think it like those two fields coming together made a beautiful paper.
I agree. Yeah, this is exciting stuff, and it shows this. I guess what E.O. Wilson would call consilience. It's like this wonderful like intermeshing of these different fields, where you know paleontologists and evolutionary biologists and geneticists and ichthyologists can come together and make discoveries that kind of draw upon all of our different areas of expertise. It's inspiring in a way, and I'm excited about it.
I want more, but only because I thought it was so cool.
Yeah, I agree. Top-notch paper. I would have loved to have seen this in a higher profile journal. I was kind of surprised that I'm not. You know, I have colleagues who have published in Evolution before, but it's not a journal that I see a lot of cool papers come out in. I don't hear about it all that often.
It's historically at least been behind a really high paywall. Like to even get it open access, it was like we had been looking at one publishing when I was an undergrad, like over 15 years ago now, and it was like $5,000 to make it open access. Where a lab with the funding we had, like there was no way that you just leave it behind a paywall, and it makes it a little bit harder. I think, to me, the thing that would have put it over is if there was some level of like testing, like we talked about, if there is resilience to some kind of mutation rate, putting that in a cell and just showing it's like some kind of like
if
you've
they
done
had a
this
mechanism like that they could demonstrate. That
yeah,
would
like
have really
why,
been
right? It's like
groundbreaking. Yeah,
like they can't prove the why in the sense of the land changes or what the bottleneck was, but if it's something like on that genetic level, like if they find a cool, like I don't know, even if they find a cool gene, and they take that cool gene and just mutate it and put it into a cell, or like take the copy of one fish, that gene, and put it into the other one and show there's no difference, like even something like that I think would be next level. But it's much easier said than done. Like having any kind of CRISPR genetic engineering sounds very easy on principle, but it is not actually easy. Like you have to adapt it for every single animal that you do. The injection protocol
doesn't sound easy to me. Shoot.
Well, like easy in like, you take your guide RNA, you throw that in, you put in some Cas9 that'll chop it up, that puts in the new gene you want, boom, you're Gucci. And my professor would always be like, "Oh, this is the easiest thing ever. Like, just do it. What are you waiting for?" But it's very much like the concentrations to make that theory work. The concentration, the timing, the growing up the animal. It just is an exorbitant amount of time. That's why maybe I thought a cell line, like putting it in a cell, like a gene of interest, and seeing like if you radiate the cells, is the repair mechanism work better? Maybe, you know, like those kinds of not easy ideas, but maybe they're slightly easier than working with the whole organism.
Yeah. Huh. This has been a lot of fun.
Yeah. I know it is late for you, my friend. We got totally rabbit hole here, but I hope it was I hope you enjoyed it as much as I did, and chat you as well.
This is great. Yeah, shoot, I'm glad we independently found such a cool paper, and I'm eager to read this cover to cover now because I didn't have a chance to yet. But yeah, I feel like I'm going to be talking about this paper for a while with colleagues and friends, and it's good stuff. It's good stuff.
Oh, and Uwu's
I feel like
Lab.
we're going to be returning to this. We're probably going to be referring to this in the future, I imagine. Yeah.
Yeah, and Uwu's Lab is here too, so thank you for the discussion. Uwu, thank you for being here.
Yeah, hey, well, welcome.
Yeah, it's a really fun paper, and just thank you, Danny, for the time. I assume, my friend, you should probably get some dinner, right?
Yeah, it's about that time. But yeah. Anyway, thank you for the chance to discuss this, Glenn.
Of course. Thank you, my friend. Is
wonderful
there anything we can promote? Of course, let's try pinning another message. See if my pin is no longer broken.
Shoot.
For Danny's field work, y'all, these are supplies that you can contribute off this Amazon list. It's
my crew.
Anonymous, like it doesn't go in, like you don't see Danny's address, it doesn't dox him, but it's
it's
great.
also not my address. It gets shipped to Utah, where Ethan lives. Because I'm gonna meet him in Utah, and then we're gonna drive up to Wyoming at the end of May, and we're gonna be digging up at least 2 new species of dinosaur in Wyoming, probably more, if we're being honest. But yeah, any dinosaurs that we find in that formation are pretty much guaranteed to be new species or new genera. And I'm doing my best not to be too modest about this. You know, there's this inclination that we have as scientists where it's like you never want to be too, emphatic about the work that you're doing or that sort of thing. But I've got to like fight against that instinct to be like, no, this actually is really cool and really important. Yeah, we're going to be doing neat stuff out there, and if people want to contribute in a direct material way, that's an easy way to do it, is with that wish list.
And also to mention, you'll be live streaming it, which I don't know of anywhere else in the world that you can see someone with the expertise that Danny has digging up dinosaurs and telling you about them and taking you along with that journey. Like, this man right here, Chad, he's not going to say it, so I'll say it. He's inspiring a next generation of scientists. And thank you for everything you do. Like, that field stuff is honestly a game changer for the STEM community as a whole.
I appreciate you saying that, Glenn. Shoot, you're gonna give me a big head.
No, I'm serious. Like, y'all fell in love with science from dinosaurs, and having someone to be live in the field and teach you about the dinosaurs and how to dig them, and you could ask questions and get like live reaction and feedback. That's nothing to just like poo-poo. That's, Danny, that is huge, and you don't have to ever do it, and you do. And I know it's your full-time gig, and I just want to say how much it means to so many people of what you do.
Well, it's my privilege to be able to do that. I mean, it would not be possible without the support of everybody, all the viewers on my channel, supporters. I feel like the luckiest guy in the world to be able to do that. Something I kind of dreamed of since I was a kid, and now I get to share that with everybody, and I just feel incredibly lucky. So, yeah.
Yeah, if y'all see the heart button right now at the top of the chat, that means you're not following Danny. Please fix that by pressing the heart button. Easy.
And Likewise, Belinda, thank you for, again, providing this platform for science outreach and taking the time to read these papers and listen to me yammer on about this stuff. The work that you're doing here, of course, is pretty incredible. I mean, the chance that people have to ask a geneticist about genes is pretty darn cool. I mean, genetic technology is one of those things that this is going to change the world for the next century. And you're here on the cutting edge of that, and you're breaking down these concepts for everybody watching. I hope people realize how special this opportunity is to be able to ask you questions. And thank you for doing what you do here, because it definitely does clearly make a big difference to people who are watching. And the community that you've built here is impressive. And it's, I don't know, I feel privileged to be able to be here and try and do science outreach alongside you.
Right back at you, Danny. And y'all, we had a panel at TwitchCon together. We got to share the stage last year, and hopefully we'll be doing something again this year.
Yes.
And not confirmed, Danny. Not confirmed, but Lita and the Munchkin might come to TwitchCon, and so we might be able to.
Talking about that lately, yeah. Very exciting. It'd be lovely to meet Lita and baby Alona. Yeah, how are you going to manage that with a baby? Holy cow.
Well,
it's going to take some work.
Yeah, and if we've talked to them, like there are actually friendos in the community who will be like at TwitchCon, and so we're like, if we get a panel, it'd be great to pull Lita on too, and one of our friends can hold the Munchkin.
Yeah. Or maybe she can have a microphone too. I don't know.
Oh, I mean, yeah, she has opinions. That's fair.
Definitely, yeah.
Before you go, Danny, is there anything else that you'd like to promo? Upcoming things? I know it's Thursday, Burbs Day tomorrow, but always, my friend. There's anything in the world.
I need to hear back from Creatrix, but she's probably replied to my email already. But we're gonna, I think, be having a special stream about bilbies, an Easter bilby stream. Australians in the chat probably are already aware of the Easter bilby. There's this wonderful marsupial mammal in Australia called the bilby. It looks kind of like a marsupial rabbit. Rabbits are invasive in Australia, and so the idea of like the Easter bunny is, I don't know, it kind of is. Rabbits are like to Australians a symbol of like ecological devastation. They're an invasive species. They're not a good thing to have around, and so a group of conservationists in Australia came up with this idea of, instead of a chocolate rabbit for Easter, having a chocolate bilby. And some of the money that goes from the sale of these chocolate bilbies goes toward the conservation of the actual bilby animal. And we're going to be talking about that, kind of celebrating how cool marsupials are and how unique the Australian continent is with its fauna. And I'm going to be 3D modeling and printing a bilby for this. So, yeah, that might be on Friday. We'll see.
Nice.
So I'm excited about that. It's gonna be good.
Awesome. Yeah, we just shouted out Britt for anyone who's interested. She's been leading a charge on it. Her TikTok has been
Together,
a bunch of different bilbies,
galaxy.
different models. And Hip, thank you for the resub. Another Partner Plus point.
Spectacular Raptor just subscribed for 11 months. Always learning new things here, and I love it. Thank you all.
And a piece of raw science. Thank you for being with us, Hip, and supporting science on Twitch. We are taking the platform by storm. I don't care what people say. Soon it won't be just for gaming. People will think like, oh yeah, I learned stuff on Twitch, and that'll become much more normal. It won't be the weird that it's gaming. It's gaming, right? It's the game. It'll be like, no, you can learn stuff here. And
Twitch.tv. That's the science place, right? That's where I go to to learn about things outside of school and have fun.
Yeah.
Yeah.
So again, thank you so much, Danny. We'll see you tomorrow on your stream. I'm looking forward to always Thursday, Burbs Day, and the Bilby stream is going to be super cool. Ask Britt. She posted something on her Twitter with a 3D printed chocolate Bilby as well. So there's been a ton of variety of them too.
Nice. Cool. Well, yeah, shoot. I need to check my email and see if she's gotten back to me. I've been a little neglectful about that, how busy I've been. But I will see you later, Belen.
Yes,
Thanks
sir.
so much for everything, and take care, chat. You all have a good evening.
Bye,
We'll see you
Danny.
later. Bye-bye.
All right, y'all. That was the lovely Professor Anduza of Paleontologizing.